In this session, we are going to learn about some of the early experiments conducted by different scientists that led to the discovery of overall process of photosynthesis. So let us start with the first experiment, which was conducted by Joseph Priestley in 1770. Now the main contribution of his experiment was that he proved the role of air in the growth of green plants. Let us understand Priestley's bell jar experiment with the help of the diagram. So, he took a bell jar and he put a burning candle inside the bell jar, and also he kept a mouse inside the bell jar, and the bell jar was tightly closed. After some time, he observed that the candle got extinguished inside the bell jar, and the mouse which was kept inside the bell jar also died after some time. So he concluded that the candle and the mouse were releasing something that was damaging the air. So whatever air was present inside the bell jar, that air got damaged because the burning candle was releasing some chemical, some gas, and the mouse also was releasing some kind of a gas. What was that substance or gas was not verified at that time. Then what he did, he placed a mint plant along with the burning candle and the mouse inside the bell jar. So he used a mint plant this time. And he kept the mint plant inside the bell jar along with burning candle and the mouse. After some time, he observed that the candle continued to burn. The candle did not extinguish. Also, the mouse stayed alive. The mouse did not die, as in the case of the first time. So, he concluded that plants restore to air whatever breathing animals and burning candles remove. So, because there was a presence of mint plant this time along with burning candle and mouse, he concluded that this plant restored to the air what was taken up by the breathing animal and the burning candle. So whatever was being released by the burning candle and the mouse that was damaging the air, that is also taken up by the plant. Along with that, plant was releasing something. Plant was releasing something which was taken up by the burning candle and the mouse, because of which the candle continued to burn and the mouse stayed alive. Why did the candle not extinguished, or why did the mouse did not die here? Because the plant released that substance which was necessary for the candle to continue burning and for the mouse to survive. Right? So this was later discovered that it was oxygen which was being released by this mint plant that helped in the combustion of candle and the survival of mouse inside the bell jar. Alright, so this was his experiment in 1770, and later in 1774 Priestley discovered oxygen. So he later on in 1774 discovered oxygen, and it was oxygen only which was released by the mint plant that was helping the candle to continue burning and the mouse to stay alive. Alright, so this was the Joseph Priestley's experiment. I hope it is clear. He explained the role of air in the growth of green plants. In a way, oxygen is present in the air, in the atmosphere, right? So he indirectly kind of demonstrated that oxygen is released by the green plants. All right. So let us now move on to the next experiment, which is Jan Ingenhousz's experiment. Now, the main conclusion of his experiment was that sunlight is essential for photosynthesis. Jan Ingenhousz, he proved that sunlight is essential for photosynthesis. So what he did, he just repeated what Priestley did in his experiment, but in the presence of light and then in the absence of light. So he just repeated those experiments once in light and once in dark condition. So what he observed that this oxygen or whatever was being released by the mint plant, it only it was only released when there was a presence of light. Alright, so he utilized the same apparatus and he kept the burning candle and the mouse inside the bell jar along with the mint plant. Once he kept it in light and once he kept it in the dark. So when kept in the dark, the mouse died and the candle extinguished. Why? Because in the absence of light, in dark condition, the plant was not able to photosynthesize and hence no oxygen was released. Therefore the mouse died and the candle stopped burning. But in the presence of light, it did not happen. In the presence of light, the candle continued to burn and the mouse stayed alive. Why? Because in presence of light, photosynthesis was being carried out and oxygen was being released by the mint plant. Alright, so he concluded that sunlight is essential for photosynthesis in a way, because the candle continued to burn and the mouse survived only in the presence of sunlight. Alright, and he performed one more experiment utilizing an aquatic plant. So what he did, he took an aquatic plant and kept it in a tank of water. So this is the aquatic plant, green plant, and he kept it in a tank of water, and in the presence of light he kept the apparatus. So after some time, what happened? He observed that there were small bubbles that were arising from the green parts of the plant. So what could be causing the arising of bubbles around the green parts? So he observed that small bubbles were being formed around the green parts in the presence of light. And later he discovered that these bubbles were of oxygen. And on the basis of that, he concluded that only the green parts could release oxygen. Only the green parts of the plant could release oxygen. Alright. So, using this aquatic plant, when the aquatic plant was kept inside the tank and kept in the presence of light, then all the green parts of the plant showed presence of or accumulation of small bubbles around the green parts. That means those green parts were performing photosynthesis in the presence of light and oxygen was being released, right? And it was proved to be oxygen only later. So it was Jan Ingenhousz only that discovered that these bubbles were of oxygen, and on the basis of that it was concluded that only the green parts could release oxygen. Only the green parts of the plant during photosynthesis releases oxygen. Alright, so this was the experiment of Jan Ingenhousz, proving that sunlight is essential for photosynthesis, as well as only the green parts of the plant could release oxygen. Moving on to the next experiment, which was of Julius von Sachs. So Julius von Sachs in 1854, with the help of his experiment, concluded that glucose is produced in the green parts of the plant. And the glucose which was produced in the green parts is stored in the form of starch. Alright? So the production of glucose during photosynthesis in the green parts of the plant And the storage of glucose in the form of starch was proved by Julius von Sachs. He also demonstrated that the green substance, which we now call chlorophyll, is located in a special bodies which we now call chloroplast inside the plant cells. So the exact term chlorophyll and the chloroplast were not identified at that time, but he somehow discovered that there are green substances which are located in the special bodies inside the plant cells, in which during photosynthesis the glucose was being produced and stored in the form of starch. Alright, so all these major discoveries were conducted by Julius von Sachs in 1854. Then the next experiment related to photosynthesis was performed by T.W. Engelmann. So he demonstrated the first action spectrum of photosynthesis. Now what is action spectrum? We studied about action spectrum and absorption spectrum in the lecture in which we covered the photosynthetic pigments, right? So if you are new to this channel, or if you are watching this video for the first time, you can check out the lecture about the photosynthetic pigments. Or if you have forgotten and need to revise what is action spectrum and absorption spectrum of photosynthesis, you can revisit that lecture again. So the experiment conducted by T.W. Engelmann, let us understand with the help of this diagram. So what he did, first of all, he took a green algae called cladophora for this experiment. So he took cladophora, which is an aquatic algae, green algae, and he put it in a tank, and he exposed this green algae to the visible spectrum of the light, the visible spectrum. So how he exposed the, if you remember the splitting of light, if you remember that if we pass the sunlight through a prism, then it split into its constituent 7 colors, which is the Vibgyor, right? So Engelmann did exactly that. He utilized a prism, and then with the help of prism he split the visible light or sunlight into 7 colors, Vibgyor, and then he exposed the green algae cladophora to this split spectrum of 7 colors. Now he also added some aerobic bacteria in the water tank containing cladophora. So after some time, what he observed, that the aerobic bacteria started accumulating in the region of blue light and red light. Alright, so what does it prove? What is causing the aerobic bacteria to accumulate in the region of blue light and red light? That means oxygen must be releasing more in amount in the blue light and the red light region, because these are aerobic bacteria, and we know that aerobic bacteria require oxygen for their survival, right? So they will be accumulating in those regions where oxygen is being produced. And we know that the other experiments, the earlier experiments, proved that oxygen is released during photosynthesis, right? If you remember that experiment done earlier, which was kind of similar to this experiment, when the small bubbles of oxygens were produced. So similarly, here also oxygen was being released from all the green parts of the plants, but more towards the blue region and the red region. That means the rate of photosynthesis is maximum in the region of blue and red light. That is why it is the first demonstration of action spectrum of photosynthesis, and it is also kind of similar to the absorption spectrum of chlorophyll A and chlorophyll B that we studied in the lecture about photosynthetic pigments, right? So this was about the experiment done by T.W. Engelmann. Now, moving on to the final experiment which was done by Cornelius van Niel. Now, Cornelius van Niel proved that photosynthesis is a light-dependent reaction and hydrogen from a suitable oxidizable compound, which is water in the case of photosynthesis, reduces carbon dioxide to carbohydrates. Now, what does it mean? We know that in the process of photosynthesis, carbohydrates are produced, and carbohydrates are produced with the reduction of carbon dioxide. Reduction means addition of hydrogen. So where that hydrogen is coming from? Cornelius van Niel proved that this hydrogen is coming from the water. So water is the oxidizable compound which is reducing the carbon dioxide to carbohydrates. Alright, so in case of green plants, it is the water which is the hydrogen donor, and water donates the hydrogen and reduces carbon dioxide to carbohydrates, and itself gets oxidized to form oxygen. So if we look at this chemical reaction here, the water here is giving its hydrogen to carbon dioxide and reducing the carbon dioxide to form glucose C6H12O6, and the oxygen from the water is evolving as oxygen gas. So it is the hydrogen from this water that is reducing the carbon dioxide and itself is getting oxidized to form oxygen. This is true for the case of green plants during the photosynthesis process. Now, what about the other organisms that do not utilize water but utilize some other chemical compounds like H2S? So in case of the green and purple sulfur bacteria, it is the H2S or hydrogen sulfide which is the hydrogen donor and not the H2O. Also, in this case, oxygen will not be released. Instead, sulfur or sulfate compound will be formed depending on the type of organism. Alright, so this is the chemical equation when the green and the purple sulfur bacteria carry out photosynthesis with the help of hydrogen sulfide. So carbon dioxide combining with H2S. So the hydrogen from the H2S will combine with carbon dioxide and reduce the carbon dioxide to form glucose or carbohydrate C6H12O6, and the sulfur will be oxidized and form a free sulfur gas released. Alright, so here in this case, the hydrogen is coming from, not from the water but from hydrogen sulfide. Hydrogen sulfide is the hydrogen donor here. So in case of photosynthesis, we can conclude that the oxygen comes from the water and not from the CO2. Because CO2 and H2O both contain oxygen. So initially scientists were confused that the oxygen which is evolved in the process of photosynthesis is coming from carbon dioxide or water. So this confusion was clarified with the experiment of Cornelius van Niel, right? So he concluded that the oxygen comes from the H2O and not from CO2, and this was later confirmed by radioisotopic studies as well. So if the oxygen was coming from carbon dioxide, then some other product would be formed here, right? And with the help of radioisotopic techniques, it was later confirmed that it was water which is donating its oxygen, or the oxygen is coming from the water and not from CO2. So what is a radioisotopic technique? For example, 6 CO2 plus 12 H2O. So here in this case, the oxygen we will use for water, this oxygen will be a radioisotope. Okay, so I'm putting an asterisk mark here, which proves that the oxygen of the water is radioisotopic. Now, in the process of photosynthesis, we are using a radioisotope in the water that is being used in the photosynthesis. And now the C6H12O6 carbohydrate will be formed and oxygen will be released here. Alright, I'm not balancing the equation so that we can focus on the radioisotopic technique. So what happens, this oxygen which is released here, it was tested and found that it was the same radioisotopic oxygen which was utilized in the, which was present in the water that we used for photosynthesis, right? If the oxygen from CO2 was being liberated, then we would not find the radioisotopic oxygen to be released in the photosynthesis process. Then the normal oxygen would be released because we are using a normal CO2. The oxygen in the CO2 is the normal, which is not radioisotope. So that normal CO2 would be released, right? But it was not found to be like that. The oxygen which was released in this experiment, in which we used a radioisotopic oxygen inside the water, the oxygen which was released, it was radioisotopic. It is identified using this different technique that this oxygen was radioisotopic. So that proves that this radioisotopic oxygen, the source can only be this radioisotopic water only, not the CO2. Right? So it confirms that the oxygen which is released in photosynthesis comes from the water and not CO2. So I hope this is clear how with the help of radioisotopic technique it was confirmed that oxygen comes from water and not CO2. And so after all these experiments, a chemical formula for the photosynthesis was derived, a full chemical formula, and which is this: 6 CO2 plus 12 H2O in presence of light will give C6H12O6, which is your glucose or carbohydrate. This will be stored as starch in the plants. Then 6 H2O molecules will be produced and 6 molecules of oxygen will be produced, which is released in the atmosphere. We know that green plants, they release oxygen during photosynthesis, right? And they utilize carbon dioxide from the atmosphere. So based on all the data gathered over the year by various experiments conducted by different scientists, led to the discovery of this full-fledged chemical reaction of photosynthesis. So this chemical reaction is important. It might come in your exam to balance out this chemical reaction. So please remember the number of molecules present in each of these raw materials and the products of photosynthesis. Alright, so this was all about the early experiments related to photosynthesis. So let us have a quick summary of all the experiments that we studied. The first experiment was conducted by Joseph Priestley in 1770, in which he proved the role of air in the growth of green plants, and he concluded that plants restore to air whatever breathing animals and burning candles remove. He indirectly kind of concluded or discovered the importance of oxygen, that oxygen is being released during photosynthesis. and he later on discovered oxygen in 1774. Then the second experiment was of Jan Ingenhousz, which proved that sunlight is essential for photosynthesis, right? And he utilized a green aquatic plant, and in the presence of sunlight he saw that small bubbles were being produced, and these bubbles were later discovered by him as oxygen. He also proved that only the green parts could release oxygen. Then Julius von Sachs proved that glucose is produced during photosynthesis inside the green parts of the plant, and it is stored as starch. He also indirectly confirmed the presence of green substances in special bodies, and he was talking about chlorophyll and chloroplasts, which were not discovered that time. Then came the experiment of T.W. Engelmann, which kind of firstly described the action as well as absorption spectrum of photosynthesis. He utilized a green alga cladophora in a water tank containing suspension of aerobic bacteria. He also used the prism to split the visible light into its spectral components and then illuminated the green alga with this Vibgyor light, the visible spectrum. And he observed that the most amount of photosynthesis was occurring in the blue and the red region of the visible light spectrum. How he concluded that? Because the aerobic bacteria started accumulating more in those regions. So the first action spectrum of photosynthesis as well as the first absorption spectrum of photosynthesis was also kind of demonstrated by him. Then the experiment by Cornelius van Niel proved that photosynthesis is a light-dependent reaction. Hydrogen from a suitable oxidizable compound, which is water, reduces carbon dioxide to carbohydrates. In other words, we can say that in the cases of green plants, H2O is the hydrogen donor, and the oxygen which is released in the process of photosynthesis comes from water and not CO2. And this exact thing was later verified by radioisotopic techniques. In the case of green and purple sulfur bacteria, H2S is the hydrogen donor and not the water. And instead of oxygen, sulfur or sulfate is released. Alright, and on the basis of all of these experiments and the data collected over the years, a complete balanced chemical equation of photosynthesis was discovered, which is this, enclosed in the box. Alright, so I hope each and everything is clear to you. That's all for this session. Thank you so much.